Okay, ladies and gentlemen, I will show you the comprehensive SERS platform for biomedicine and food safety detection in International Conference on Sensing Technology of Science Forum. This is Jianzhong Zhang from the National Zhongxin University of the Taiwan. Okay, I will show you the signal enhancement chip based on the three-dimensional plasmonic hotspot. So we use this chip to be combined with a portable Raman spectroscopy to construct a signal enhancement system. We use this system to collect, analyze, and build up the spectrum database, and then we analyzes the database to become the barcode system and transfer to be a big data library like this. And finally, we build up a database we can detect and clinical testing for any analyze even production. So now we have successful on the agriculture and biomedicine application. And this system, I will show you the chip. We construct a three-dimension random cross-wire wood type system. The narrow chip is three-dimension narrow wire construct layer by layer like this. So there are so many cross sections between narrow wire like this. And there are so many plasma conjugates like this. So in this chip, in this system, and this three-dimension chips, the local surface plasma resonance is isotropic. So they can enhance the signal in any place of the cubic. And the plasma enhanced is homogeneous. So that's why this chip can detect any size of analyze. So finally, this chip can enhance more than 100,000 times of the Raman signal. Okay, but in the first stage, we need to find the optimized three-dimension RCW condition. For example, detection of limits and the overall equilibrium, effectiveness, and stability. We use the standard Rodamin to detect the detection limit of this three-dimension RCW chip. And we find the detection limit can be the picomolar concentration based on the 1 second collection. And then the IEE error does not exceed 5%. This is a very fine reproducibility. Okay, and then the nanostructure chip can be stored more than 80 days under normal condition without packing. So it's very important because all the SERS chip was constructed by a noble metal. So it's very easy to be oxidation or rust. So it means our chip is very stable and it keeps more than 18 days. Okay, till now we have collected so many field data. For example, we can successfully collect the drug signal, and we can identify and detect the purity of the drug. On the other hand, we can recognize the cancer in the normal cell based on the SERS signal. And we can also detect the bacteria. For example, the MRSA, the positive, gram positive bacteria, and the gram negative bacteria, E. coli. We can also collect coronavirus like this, and we use this tag to collect the archip, the plant virus with high academic plans like this. So this is a successful case. I want to show you a special case. It's recently we successful to finish the clinical test to detect direct bilirubin with cooperation with NTU hospital. For children, the direct bilirubin is a very important issue. However, for adults, the direct bilirubin recently was reported have positive relationship with COVID-19 syndrome. So, however, we have success test more than 20 patients, okay, which match very well with the CV result of the hospital. But more important, our test time is not more than 5 minutes. So in the first step, we construct a standard and build up a correlation shapes between the concentration and the signal. So it's easy to find the patient. We just use the serum with very small volume of quality. And then under this clinical test, I will show you how we face the problem and how we dissolve this problem, especially on actual application and the clinical field. This is a real test. We collect spectrum, but with a very complicated and very pattern background. Sorry. And how we use our AI system to resolve, dissolve, and deconvolution, and build up the fine spectrum system, and then to construct the AI and the barcode systems. So first step, we need to collect more and more data, and then we need to smooth and remove the background noise. So we use the CWMA, Central Web Moving Average, and the program to construct and build up a very beautiful, or I will say is a normal spectrum. And then the spectrum analysis is deep neural network to recognize the major components. Finally, we use the LP neural network model to build up the barcode. So till now, we can collect the spectrum, and then we can build a barcode system to build up the big data for the library. Okay, I will show the example is the pesticide. It's very important in agriculture. So now we have built up more than 10 kind of pesticides, and to clean and to build up the fingerprints of the spectrum, and then finally we build up the barcode as I have introduced the method. So it's very successful from the detect to build up the data and the clinical analyze to construct the big data or cloud data. So it's a very successful case. Now we are trying to collect the COVID-19. The COVID-19 virus, coronavirus, and the first steps, we build up the spike protein database. Under this condition, we need to find the optimal condition for our chip, especially what kind of lacing is very important. And finally, we find the 785 nanometer lacing is more suitable because all the biomolecular is very easy to decompose and very easy disappear by very high power lacing. So we spend so many time, waste so many time to test and find the optimal condition to test the biomolecular, especially the spike proteins. On the other hand, it's more important we detect the antibody. The antibody of the spike protein is successful construct like this. So what is our purpose? We want to detect the antibody signal on the broad. So we have realized the broad, the antibody in broad is more important. It's more important to detect your antibody in your human body more than you detect the virus in your body. So now we have success, but next step we want to collect more and more data and to construct the big data. So I will conclude our three-dimensional RCW chip. The spirality is pre-treatment free. We don't need to prepare in advance. And then the detection is directly. We don't need to conjugate the antibody. So it's an antibody-free chip. And the total detection time is less than 5 minutes. And our system is solid-state detection. So there's no liquid spores issue problems. And our detection volume is 20 microliter. It's a microanalyze. And our chip, it can be used in any Raman equipment. So don't train in specialist. So I'll conclude by this diagram, this chip, because he is LSPR isotropic and their plasma is homogeneous. So we can detect any size of analyze. Especially as I mark here, we are focused on the biomolecular detection and we success. Okay, so I will thank the National Science and Technology Council of the Taiwan and thanks the NCW and behalf of the Smart Agriculture Research Center team, I will thank you for your listening. Thank you.